thank you for the kind introduction. Thank you for showing up in such big numbers. I'm going to take the next 10 to 12 minutes to walk you through what I now call the evolution of complexity in proteins, no longer just the evolution of complexity in Rubisco, but it is, I guess, very Rubisco specific. And so I started out my PhD with the main observation that many proteins assemble into obligate heterooligomers. What I mean by that is a protein complex that needs more than one subunit to come together in order to form a functional quaternary structure. A simple example of this could be Rubisco, which just needs two distinct subunits that all have to find each other in order for the protein complex to be functional. A more elaborate example, of which there's many out there in nature, would, for example, be ATP synthase, where 17 distinct subunits all have to come together in order to form the active protein complex. And if you take any of the subunits that constitute ATP synthase away, this protein complex wouldn't be functional anymore. And now the interesting thing about this is that many of these complexes actually evolved from less complex ancestors. So if you look at the genome of, for example, prokaryotes and compare them to the genomes of eukaryotes, you often find that homologous protein complexes basically exist in simplified versions in those prokaryotic organisms. So at some point over the course of evolution, there must have been an evolutionary interval over which an ancestral form of a protein complex gained a new subunit and basically evolved both an interaction on this new subunit as well as a dependence on it, in order to then form this obligate heterooligomer. And now what makes these obligate heterooligomers difficult to work with as biochemists is that these newly recruited subunits are often of unknown function, right? This protein complex carries out seemingly the same function. It now has a new subunit. And the problem is, if you take that subunit away, this protein complex just isn't functional anymore. And me, speaking for myself as a biochemist, I would like to just be able to take that subunit out and see what it does to the protein complex. So starting out my PhD, I wanted to investigate how we can study the function of such essential subunits in protein complexes and why proteins actually start to depend on newly recruited subunits over the course of their evolution. And I tried to tackle this project by taking a protein complex, in this case Rubisco, and trying to reconstruct its evolution as a biochemist in the lab, in the wet lab. Rubisco as the model system, very briefly. Rubisco is a carboxylase that carboxylates ribulose 1,5-bisphosphate, and it's the key enzyme of the Calvin cycle in plants, algae, cyanobacteria, most photosynthetic organisms out there in nature. It also has a side reaction with oxygen. This is an undesired side reaction where it, again, confuses oxygen for CO2 and produces a molecule called 2-phosphoglycolate, which has to be detoxified in an ATP and CO2 releasing side reaction or biosynthetic pathway. And sort of the quality with which Rubiscos can discriminate between these two gases is called their specificity factor. If you look out into nature, this is a picture of Marburg, the place that I went back to for my PhD, you will find Rubisco anywhere. Rubisco is in all plants, algae, cyanobacteria, everything that's photosynthetic out in nature. And in most of those organisms that you can see by eye, Rubisco forms a complex that looks like this. So you have 8 large subunits, here in light and dark gray, arranged in a cyclic assembly, with 4 small subunits at the top and 4 small subunits at the bottom of the complex. The small subunit in Rubisco is non-catalytic. In fact, it's not even close to the active site. It's about 25 angstroms away, and people don't really know what it does yet. Even though Rubisco is a very well-researched protein, people don't really know what the small subunit does, because it is of the nature that if you take it away, the protein complex is not functional anymore. Yet it's also an evolutionary novelty, and this is what makes it interesting for us to study. So if you look at sort of like a phylogeny here in a schematic form, you will find many, many organisms. These are all bacterial or archaeal, that encode Rubiscos that don't have a small subunit gene at all. And when you purify their Rubiscos, they form mostly dimers of large subunit, but they can also form larger oligomeric structures of just large subunits of Rubisco. And then there's one single evolutionary interval, after which all the descendants of that interval form this L8S8 assembly, so these 8 large and 8 small subunits that come together in one complex. The only thing that people sort of were able to think that the small subunit does is that it might improve specificity, but this is purely based on the observation that all of these Rubiscos that contain a small subunit have comparatively high specificity, whereas all of the ones that do not have a small subunit have comparatively low specificity. And we now sought to see whether or not we can answer this by trying to retrace this evolutionary interval to see what the small subunit does. Basically trying to answer whether or not the small subunit fine-tunes catalysis. For this, I'm now in the next slide going to zoom in onto just this evolutionary interval up here, which is this, where at the start of my PhD, I discovered 3 new clades that sort of break up this evolutionary interval and get us a bit closer to these L8S8 Rubiscos, these Rubiscos composed of a large and a small subunit. taking these into the lab, purifying them and analyzing their structure, I found that some of these clades already encode Rubiscos that form oligomeric assemblies of essentially octameric assemblies, yet they still don't encode a small subunit. So now I could have done one thing, which is take one of these Rubiscos and take one of these Rubiscos and just compare them to one another to try to figure out what the small subunit actually does. The problem is these are still sort of 250 amino acid mutations separated from each other. So it's very difficult to actually pinpoint any sort of given phenotypic change to specifically the small subunit and not to any of the 250 amino acid changes between those two proteins. And so what I decided to do instead is I decided to use an algorithm called ancestral sequence reconstruction. What this basically does is it turns the algorithm that is used to build such a phylogeny on its head and asks, okay, what is the most likely sequence of a protein that would have existed at an internal node of this phylogeny? So right here, to explain the distribution of all the sequences that exist now at the tips of this phylogeny. So this is the best statistical estimate given the observation of all the sequences that we have that exist in nature. With this, I resurrected two large subunits, one that we think existed right before the small subunit first evolved, and one that we think existed right after we think the small subunit first evolved. Alongside that, I also did the same analysis for the small subunit and basically reconstructed the oldest possible small subunit. And then basically got them into the wet lab and analyzed them. First thing I did is just look at their structure. What you're seeing here is a mass photometry data. Essentially, this is just a histogram of the protein complex mass in solution. And this is a crystal structure. So we basically see that UNC-L, this protein down here, is just an octamer of large subunits, as we expected it to be. And then going up the evolutionary interval and pairing it with the small subunit, we see that that ancestral Rubisco now forms this L8S8 complex. It interacts with the small subunit and essentially forms this hexadecameric protein complex. Now, first thing we wanted to ask is, okay, now, does the specificity of Rubisco improve along this interval? Long story short, it did. It doubled. Great. So this could be due to the small subunit, but there's still 95 substitutions that occur along this evolutionary interval. So I was like, okay, maybe by chance, this protein down here does not yet actually need the small subunit, but can interact with it. This didn't turn out to be the case, so whatever I tried, I couldn't get it to interact with the small subunit. But then alternatively, maybe this protein up here doesn't actually depend on the small subunit yet. Similarly here, whenever I took the small subunit out, I wouldn't get any soluble protein anymore. It already strictly depended on this new subunit. These proteins, as I already mentioned, are separated by 95 substitutions. And in those 95 substitutions, we seem to have gained the interaction between those two subunits, but we've also seemed to have accumulated all the substitutions that actually make these protein components depend on each other. So now I asked, well, can I use the evolutionary information that I have, the sequences and the structures, to actually sort of reconstruct this evolutionary interval to see if I can get a protein variant that can bind the small subunit but doesn't need it? So looking at these crystal structures, I could see that 14 of the substitutions that occurred along this evolutionary interval are in the interface. And then when you have a closer look, only 7 of those actually actively engage in this interaction between the large and the small subunit. I then decided to basically transplant these substitutions into UNC-L. So if an alanine changes to a glycine, I will take the glycine and put a point mutant into UNC-L. With this, I created a 7-mutant construct, which I termed UNC-L plus 7, which was highly soluble on its own, functional and everything. It behaved very well. And then when I added small subunit, it could bind the small subunit, and we basically re-engineered the evolution of that interaction. Crystal structures of this once again showed exactly what we wanted. So we have an octamer of large subunits. We can add the small subunit, and it forms this hexadecameric L8S8 complex. And now with this, for the first time, we could ask, okay, what's the function of the small subunit? Well, first thing we did is look at crystal structures, and it looks identical. Like, you have a new subunit, fine, but beyond the new subunit, everything basically looks the same. However, we still asked, okay, well, does it influence the specificity factor in any way? And so doing this assay, we see that even going from UNC-L to UNC-L plus 7 without the small subunit present, we see a slight improvement in specificity. And then adding the small subunit, we see another improvement in specificity, but none of this was really dramatic. And then this was pure chance. I found one more substitution that also occurred along this evolutionary interval. And when I introduce that, I can make that phenotype much more drastic. This one substitution that I introduced here, when the small subunit is not present, it has no positive effect. I can measure all sort of parameters of that protein. it doesn't do anything. And now, as soon as I have the small subunit bound as a sort of epistatic modifier, suddenly this one substitution is beneficial to the protein, and it actually makes it function better. So retracing sort of the evolution of a protein complex can help us decipher the function of essential protein components. That was already cool. And then the second thing we asked is why Rubisco or why any protein complex might start to depend on newly recruited subunits over the course of their evolution. And here we have sort of a very straightforward underlying hypothesis, which is that a new interaction might be able to modulate sort of the sequence space that is available to a protein. What I mean by that is that any given protein can evolve into a certain area of sequence space, and some areas might not be accessible, such as a mutation at the active site, whereas other mutational paths clearly are accessible to those proteins. Now, the idea is that gaining a new subunit might change what is accessible to your protein, and you can then expand into that area of sequence space, either adaptively or just neutrally drift into it. And once you end up in such an area of sequence space, you simply cannot take the new subunit away anymore. In a follow-up study, I could show that even in Rubisco, this can cause essentiality via at least 3 different biochemical mechanisms, where single, double, or triple substitutions, respectively, can cause Rubisco to depend on its small subunit for solubility, stability, and catalysis. These are different substitutions, but I can put 1 or 2 changes into Rubisco that are all historical. So it's not like I'm trying to destroy the protein complex. I'm taking something that evolved over the course of evolution, putting it in, and it can make the protein depend on the small subunit for 3 different phenotypes. Briefly showing the simplest one would be solubility. You gain a new subunit, and that interface can now become more hydrophobic than it could be in solution. Looking at that quickly, one of those historical substitutions is a serine to tryptophan. And when I do that, I get a loss of about 75% soluble protein. And in negative stain electron microscopy images, Rubisco now looks like this. So it starts forming these filamentous aggregates. Getting a cryo-EM structure of this, I can see these Rubisco octamers exactly engage in these types of oligomeric or filamentous interactions in a way such as this. This is driven by specifically this tryptophan 437 substitution, which sits at the interface. And importantly, it sits at an interface that is typically shielded by the small subunit when the small subunit is present. So as the L8 S8 complex, this interaction simply wouldn't happen anymore. So you're essentially making this interface more tolerant to hydrophobicity. This is just one of the examples of these different biochemical mechanisms, but in theory, basically we found that this sort of simple scheme can quite accurately tell you why a protein complex might become dependent on a new subunit. Right. Takeaway is complexification can open such new evolutionary trajectories. Exploring them will or can cause essentiality, and we can use these evolution-based approaches to help solve elusive structure-function problems of subunits that otherwise you wouldn't really be able to investigate. And with that, I want to thank many, many people that have been involved in all of my research and helped me along the way.